Hello, good morning. Bonjour, guten tag, ciao, ni hao, namaste, hola, aloha, konnichiwa, and for those of you from Oregon, hi. It's a great pleasure. I'm Sue Keck from the Salk Institute. It's a great pleasure today to have another one of our global IMMUNOTALK speakers. And today it's a pleasure to have Ben Youngblood come and join us. Ben is currently an associate member at the Department of Immunology at St. Jude's. He received his bachelor degree at Oregon State University. So I hope we have some people from Oregon online today. He went on, though, to do his graduate training down in Southern California in the University of California in Santa Barbara, where he made a lot of important discoveries in terms of understanding the biochemical mechanisms of how DNA methyltransferases work. He then switched gears to start his postdoc in Rafi Ahmed's lab in 2007, where he brought forward some of the leading expertise in epigenetics and made some pioneering discoveries about how CD8 T cells differentiate, was one of the first to profile the epigenetic regulation of the gene PD-1, which we know is a very important molecule. And it's through that experience that I was able to get to meet Ben, and he's just an exceptional scientist and person and friend. In 2014, he then joined St. Jude's, and his research program has been flourishing there, where he's looking at, continues to study the epigenetic regulation of T cells and applying it translationally to study children's diseases as well as chronic viral infection and cancer. And we're going to hear a lot about that today. So I'm really excited. And so Ben, he took, you know how we always ask a question to our speakers, and Ben decided to play roulette. He doesn't know what question he's going to be getting asked. And so I was going to ask you, who is the most important influential person in your scientific career? But I didn't want it to be Rafi or me. Just kidding. So here's your question. What is the trait in your personality that helped you the most in your scientific career?
The trait that helped me the most in my science, I'm charming.
Okay, so we both have big egos.
No, I don't know. And the person that inspired me the most, actually, you'll like this one. It was probably Pauling. And it was interesting, and it's because I think he set an example for humility. And so I did come from Oregon State, and one of the things that was really interesting about Linus Pauling is, even though he did a lot of his work down at Caltech, right, he set up the Pauling Institute, or his museum is actually at Oregon State, because that's where it was near and dear to him. His first appointment was from Oregon State as a chemist, and part of it was actually a bit of a romantic story too. Part of the reason why he agreed to do it there was they included his wife's work in the museum, or just like artifacts from his wife, like all the novels she got to read, she enjoyed reading, and it was really added a nice human touch to someone that we kind of all revere. So yeah, that wasn't you or Rafi. Sorry, Sue.
All right. Well, we look forward to your talk. You can go ahead and share your screen. Thanks, Ben.
Yeah. All right. And it was everyone's seeing it just fine now?
Yeah.
Okay, great. Let's shrink us up here. Okay. And there we go. And I get my laser pointer. And there we go. All right. So it was a minor change to the title. I swapped out, I got rid of the word failure in this title because I felt like I was being too pessimistic. And I think we need to start to think about how we see successes and how we can leverage them. And so, as Sue said, I did a lot of my work, my immunology work and training in Rafi's lab, and I pestered him for about 6 years, and he finally said, get out and go. No, it's actually a lot of fun, and I got to meet some amazing scientists and develop wonderful relationships. And so we took those things that we learned in Rafi's lab and we brought them to St. Jude. And we've been doing studies on mechanisms, epigenetic mechanisms of CD8 T cell differentiation and exhaustion in the setting of immunotherapy. Just disclosures. Just so we're all on the same page, I want to give a little brief background on the CD8 T cell immune response and then also use this as a way to introduce some concepts that we've learned along the way with our studies and how we're leveraging them. And so I think most people probably signing in on this call here know the CD8 T cell response, and they know that when this naive CD8 T cell encounters its cognate antigen here, it initiates a differentiation program that allows the cells to undergo this clonal expansion. And a large part of this observation is born out of Sue's work when she was in Rafi's lab, and then additional studies where what happens is you generate this massively expanded population with a heterogeneous pool of cells. A large portion of them are largely defined as these terminal effectors. They're great killers, but they're destined to die. And then there's a smaller subset that are still actually decent effectors, but they retain this multipotent capacity. And it's these cells that a fraction of them can go on to become these long-lived functional memory CD8 T cells. And it was at this point that I entered Rafi's lab, and we started discussing about the mechanisms for maintaining of properties over long periods of time in cell division. And so Rafi and I and others lifted this concept of epigenetic inheritance. And so what we were thinking about was, how does a cell here maintain the same acquired gene expression programs at this point as it's undergoing this homeostatic self-renewal? And you can also invoke other forms of cell division. And the way it works for a lot of cells is that they acquire this epigenetic mark called a DNA methylation in a CG palindrome context. You acquire it, the cell is going to undergo division, and that parental strand of DNA serves as a template as you undergo DNA synthesis for propagating that program to the newly synthesized daughter strand. And there's a methyltransferase that looks for these hemimethylated substrates and says, aha, there's an unmethylated strand or a CG that I need to methylate. And that's the essence of epigenetic inheritance and maintenance of acquired gene expression programs. And this is, of course, a repressive program, generally repressive. And so we took those concepts and we started to apply, as Sue said, we started to apply them to this process of T cell exhaustion. And at the time, the various subsets weren't fully formed or understood yet, but we knew that there were progressive changes in this state that were reinforced at some point. And so we extended this concept of epigenetic inheritance to looking at various imprinting that occurs in the exhaustion process. And one of the things we found was that actually these programs, and that's what I'm going to talk to you a little bit about today, these programs aren't just correlates of the changes, they're actually causal. And one of the things that we found is that if you can block these programs early enough in this developmental trajectory, and we're not sure exactly where it has to happen, but if you can block it early enough, you can actually stop this fate commitment decision, and you can keep the cells in a plastic state. And so they retain more of a memory-like property. And so the other thing this allowed us to do as we've been developing these concepts is what we learned is that we could be informed on the state of the T cell differentiation in an agnostic way. So but if you know enough about the epigenetic programs, you can take a cell, and this is really critical for human study, you can take a T cell, not know what its starting and ending point were, and sort of project where it's at by knowing the epigenetic state. And so we did this through a lot of DNA methylation profiling. Broadly, what we know is that the effector-like genes are fully methylated or repressed in naive T cells. The stem associated, and we'll talk about that in a bit, are poised or unmethylated. You become this this memory precursor. Everything's sort of poised, really broad strokes here, and then exhaustion or repressed. And so you can take all of this data, you can put it together, and we did this, and I'll show you how we did it. And you can develop this sort of normalized score that we call the multipotency index, and it allows you to take a T cell of unknown differentiation state and stratify it on this scale. 1 being very multipotent, which is a naive cell, and 0 being more terminally differentiated, which is more like an exhausted T cell state. And so what I want to so that's just sort of an introduction to CD8 T cell differentiation, how we think about this in the context of epigenetics. And what we're doing now is we're using these concepts and we're thinking about how we can modify the effector potential of a cell in the context of, in really simple terms here, blockers. So you block the developmental path, you block the cell from proceeding down this state of non-function or dysfunction or suppressed function, or reprogrammers. In the truest sense, can you take a terminally exhausted cell and walk it back by modulating these programs? And so what I'm going to talk to you today is about mostly about blockers, although we have a lot of, and I know a lot of groups are trying to focus on these reprogrammers. And so this is why, and I think this is why a lot of us are getting into this. And this is just an update from Jed Wolchuk's Checkmate study, where he's looking at T cell responses to various immunotherapy forms of checkpoint blockade therapy. And so the green line here is that wonderful result we learned from Jim Allison, where if you treat individuals with metastatic melanoma, these individuals would have normally succumbed. And now you have individuals that are surviving with a durable response. And that's the power of the immune system. It's not only protective, but it's protective for a long period of time. PD-1 blockade was introduced to this therapeutic modality, and it brought the progression-free survival up. Combination therapy brought it up a little bit more, but we're really kind of hitting a wall. And so I'm going to talk about checkpoint blockade, but also how we've learned from T cell differentiation in this context, and have extended to CAR T cell therapy. It's a different type of therapeutic modality, but we got some problems here that we have to address. And one of them is that actually initially when you take a CAR T cell and you infuse it, and the best case is CD19 CAR T cell, you can have pretty good responses, overall survival. But this wanes with time, and it's not just because of antigen escape. And so we have a waning functional property of the T cell that allows these individuals to no longer have this protection from the immune response. And so it's these two scenarios that we're looking at, and we're asking ourselves, how do you bring more people into this therapeutic window or this response? How do you take non-responders, convert them to responders? Or how do you extend this therapeutic response if it's in the context of CAR T cell? And so there's a lot of different mechanisms that are contributing to a failed immune response. We're focused on a lot of intrinsic properties to the CD8 T cells, and we're asking very simple developmental questions, but then we're extending them to the context of a therapy. So how are long-lived gene expression programs maintained in functional or these dysfunctional CD8 T cells that we see in these patients? And so I'm going to jump through a lot of different data here and just summarize it. I'm going to ask you to take my word for it. We can talk later. But it helps me get through the talk. So 2 important conceptual advances guiding our efforts. And this is a lot of work coming from people like Sue and Rafi and others, where we know now that mouse and human memory T cells can persist in a quiescent state, poised for long periods of time. And in humans, we're talking about 10 decades. And they do this by maintaining these acquired epigenetic programs for years after the initial antigen encounter. So epigenetic inheritance and maintenance of gene expression programs. The other thing that we've learned is that these programs are not just correlates of the developmental state and the gene expression status, but they're actually causal in reinforcing these developmental states. And I'll talk to you a little bit more about that right now. So these are some of the studies that came out over the last decade plus that from various labs that have really shown this nicely. I see Dietmar's online here. So this is a beautiful paper from Dietmar that came out just about the same time as this paper on the right that I had when I was in Rafi's lab. And what Dietmar was able to show was that if you take cells from a chronic environment and you put them into an antigen-free environment, we didn't quite know how to describe it yet, but that phenotype of exhaustion persisted. So there was an imprinting of that phenotype. And then at the same time, around the same time, I was working with Rafi, and what we had shown was that the changes in the PD-1 locus, the epigenetic changes, were reinforced, even if the antigen loads were really, really low. They didn't change, they were reinforced. This concept was elaborated on by both John Wary and Andrea Scheidinger. Andrea showed very nicely in humans in tumor settings that these early imprints in a tumor-specific T cell to form exhaustion were pretty stable. And then John showed that epigenetic stability in the form of ataxia, chromatin accessibility persisted after PD-1 blockade. And then what we did is we subsequently came along, and what my lab was able to show was that actually if you block, at least the DNA methylation, you block these programs, you can retain a lot of functionality despite chronic antigen exposure. And then of course we had a follow-up paper or another paper with Dietmar, and I won't get into this, where we showed that some of these programs are being driven by tox. So I'll tell you a little bit more about our causality experiments here. And this was one of the first experiments that we were doing to address this. We were taking wild type animals, wild type and DNMT3A conditional knockout animals. And so this is the de novo methyltransferase that provides that new epigenetic imprint. It was knocked out in the CD8 T cells. And we would infect these animals with a chronic strain of LC&V in a setting that drove really severe exhaustion. And what we found was after 2 months of infection, and this is at a really late stage in the immune response, and we set it up so the viral titers would stay really high. You could see that there was, and this is just a representative plot, but there was a larger number of antigen-specific T cells that were conditional knockout. This is just a frequency, but the absolute number was much, much higher. And you're seeing this, these cells were all PD-1 positive. And so for those of you who are not used to looking at FACS plots that are joining us, each dot here is a cell. And so this is the virus-specific CD8 T cells. So these are all specific to this epitope, and they're all expressing high levels of the PD-1 protein. So the dogma at the time was that these cells should have been turned off functionally, right? So they're expressing the inhibitory receptor. They should not be functional. So we tested that by asking if they could make cytokines. And so we exposed them to their cognate peptides for about 5 hours and asked if they indeed did make cytokine. The wild type were, as we all knew, would be suppressed. They couldn't make gamma, and they sure as heck couldn't make co-produce IL-2, but the knockout were just fine. The knockout can make interferon gamma and even co-produce IL-2. So despite expressing PD-1, they did not have a functional definition of exhaustion. So we went on further. Once we saw this, we went on further to ask, well, are they now responsive to checkpoint blockade? Because we knew in this model that you really started even lose functional responses to PD-1 blockade. We set the mice up with a chronic infection again, waited till you got to the severe form of exhaustion, and then we initiate anti-PD-L1 treatment. And this is what it looks like for the wild type cells without and with, and you're looking at 3 different epitopes. There's really no change, a little bit of a change in the frequency for the GP33. The GP276 expand a little bit better. You can see a bigger increase in this. And this is the one that a lot of us look at because it's a higher affinity epitope it exhausts first. And what we were shocked to see was in the setting of DNMT3A deletion, they all expanded. They all expanded massively. And in fact, if a lot of us that do mouse immunology, if you just use CD44 as a sort of a surrogate marker for the polyclonal response to LCMV, the whole doggone profile just shifts over. So it's not just the epitopes we can measure. All of these cells that are responding to LCMV are undergoing this proliferative burst. And so when Haslam was initially doing these experiments, he had to get really good because these mice would get, as you would guess, would get pretty sick. And so he got better and better at bleeding these mice and taking care of them so we could generate data like these. So this set up this concept that the ICB responsive state, and this was a few years ago now, the ICB responsive state really could be delineated by the epigenetic status of the T cells. So those cells that had this permissive state, they're permissive for effector gene expression, proliferative genes, and metabolic fitness genes. They would mount a proliferative response and maintain their effector cytokine profile, whereas these would not. So also at the time, what we were realizing was it was really, really important to get a handle on this in the settings of humans. Obviously, we want to do studies where we can cure human tumors or human chronic infections, not just mice. And so we started to profile the heck out of a lot of different human T cell settings. And so we got naive effector memory CD8 T cells from a bunch of healthy donors, or as healthy as you get in Memphis, because we all have way too much fun down on Beale Street sometimes. And then we actually had really good cohorts of HIV-infected individuals that established a bona fide state of exhaustion. And this was through a really nice collaboration with Steve Deeks, Rachel Ridishauser, and Rafik Sakali. And then we also got these functional CD8 T cells from type 1 diabetics, these autoreactive T cells, from Jerry Nepalm, from well-defined cohorts of type 1 diabetics that in some cases, even individuals had chronic disease for upwards of 20 years, but the T cells remained functional. We took all of this data, and we did the same for mouse. I'm not going to show you that now. We took all of this data, and this was really something amazing that Yiping was able to do. And he used a machine learning approach to define CPG sites across the genome and their methylation status that could roughly delineate where a T cell sat in this developmental trajectory, making with the assumption that a naive T cell was at one end of the bounds and the exhausted CD8 T cell was at the other end of the bounds. And so then he also normalized this, and this is what I told you before. This is the multipotency index, where now you know the CPG status, CPG methylation status of a cell, and you can roughly predict how multipotent or terminally differentiated it is. So at this stage, we had tools, we had concepts, and I think we were ready to ask some really tough questions. Do these epigenetic programs truly regulate human T cell exhaustion? And can we apply these epigenetic-based strategies now to enhance T cell-based immunotherapies? And so it was at this time, Stephen Gottschalk and his team were joining. He was the chair of the BMT department at St. Jude. He was joining St. Jude. And he said, hey, we can knock this out in our CAR T cell model system, and we can test these concepts with you. And so we teamed up with Steve, and really as Guidra Krenschut and Brooke Prenzing and Steve's group, and then Caitlin Zebley from my group. And we did this. We took full advantage of all the models that Steve was able to bring to St. Jude, and we tested whether DNMT3A deletion in the CAR T cell setting would preserve the proliferative function and the effector capacity of the cells in chronic antigen settings. And so the way this works is you take a T cell, pool of T cells out of an individual, and there's a wide range of developmental states. You introduce the receptor, you expand them, you grow, and you try to reinfuse. And this is really important for CAR T cell now, because as I showed you in the beginning, the efficacy for these approaches wane over time. In fact, for CD19 CAR T cells, about 2 years into treatment, there's only about 40% survival. And that's in pediatric and in some adult. So we got to get persistence going here. And so this is one of the first experiments that Guidra and her group did, was they used CRISPR to knock out DNMT3A, and there's IL-13 receptor alpha-2 CAR T cells, good deletion. And this is, again, a credit to Steve. He was realizing early on that these one-off stimulation experiments wasn't good enough. You had to chronically stimulate the T cells to sort of mimic that environment they're going into, especially with CD19 CAR T cell. And I'm showing you just one example here. This is IL-13 receptor alpha-2. But what they do now is they do the serial stimulation. And most groups have adopted this. And you can stimulate every 3 to 7 days. Here, I'm just showing you 7. And so now you're mimicking that chronic antigen exposure, and can ask what the T cells do over periods of time. And this is just a really simple readout. This is looking at the fold expansion of the CAR T cells over time. And this is why it's important to do this serial stimulation, because the CAR T cells are functional in the beginning. You can't see a difference between DNMT3A knockout and the control. Within 1-2 weeks, they look the same. They're expanding just fine. The profiles look the same. But at 3 weeks, you start to see a difference. And it's, 3 to 6 weeks. It depends on the receptor and how hard you're hitting the cells. These 3 weeks, you start to see a difference. These cells, the knockout, continue to proliferate. The wild type or the control are declining in their proliferative capacity, and the knockout continue to grow. And we've taken this experiment out to about 12 weeks without any control, of course. And so long as there's antigen present, the DNMT3A knockout CAR T cells can maintain a proliferative response. If you take antigen away, they undergo contraction, they stop proliferating. So this was broadly true. It wasn't just and it had nothing really to do with the receptor itself. We could see this response in first-generation CAR T cells, HER2, first-generation CAR T cells, HER2, second-generation CAR T cells, all the different receptors we tried. I'm showing you a plot for each individual person that we've tried this on. This is weeks on the x-axis. You really start to see this wonderful maintenance of the proliferative response only when you knock out DNMT3A, and it's highly, highly significant. And so these are the in vitro studies that led up to our in vivo test, where we would really challenge these cells now to see if they indeed could be protective. And this is one of the tougher experiments that Brooke Prenzing did. So she took an NSG animal and she said, okay, if they're really great cells, let's test them with a tough model. And so what she did was she took these NSG mice and she put in this osteosarcoma line, this LM7, and she waits now a month. And you'll see a lot of CAR T cell studies out there. They'll put their tumors in and then they right away reinfuse the T cells. No, that's not what she did. She waited a month. She let the tumors metastasize to the lung. And then she said, okay, now with 1 million cells, let's see if we can actually see some efficacy. And so indeed, that's what she saw. So here's the untreated animals. You're looking at tumor growth. The control treated animals, you see some protection, but not great. And then with the DNMT3A knockout CAR T cells, you see wonderful protection. And even when you start to see some tumor growth, it's maintained at some point unless you have antigen escape. So these were the survival curves that resulted from these studies. This is the lung metastasis model, clearly a profound survival. As I said, sometimes we got antigen escape, and that happens when you're focused on one antigen, but tremendous increase in survival. Here's some, and this is all published now. When we looked in the brain tumor model system, and this was actually one that's actually, I think this is going to be near and dear to St. Jude's heart, because this is a, there's various brain tumor these are just devastating diseases for some of these kids. And they're trying to get CAR T cells so they have extended survival in the brain. And so the model is set up like this. You do intracranial transfer of the tumor cells into the mice, and this is exactly how it's going to happen in the kids. You follow, and you actually have an intracranial transfer of the CAR T cells. There will be through a catheter. And in our model system, the tumor growth is actually quite noisy, and so this was frustrating for us. But when we saw this protection, it was satisfying. And so sometimes the tumors would go right away, and then sometimes they would take much longer to take off. But when they go, look at the slope of the curve. Boom! These suckers are taking off. They're aggressive. And when you treat it with the DNMT3A knockout T cells, sometimes it would start to take off, but you would have this maintenance of the tumor. And we waited actually quite a long time. As you can see, this is days post-injection. We took these mice down, and indeed the control, not the clearance of the tumor, but actually the control was associated with persistence of these CAR T cells at the site of the tumor. And so I think that's very important and gives a strong rationale for why we should be using these, especially in these really devastating diseases. So we wanted to understand better this mechanism, and previously we had published the DNMT3A mediated programs by doing whole genome methylation profiling in the bona fide exhaustion model system of LCMV. And so we wanted to know, is this truly exhaustion that we're looking at? And so we did whole genome methylation profiling, a lot of it. I can't tell you how much money we spent on just doing kinetics of this, but we did a lot of whole genome methylation profiling on these human CAR T cells. And really surprisingly to me, I found it very satisfying, but also surprising, comparing mouse and human LCMV versus in vitro CAR T cell stimulation, about half of the same genes, at least on the human side, half of the genes were overlapping that with the targets that we saw in the LCMV model system. So this is really teaching us that it's a conserved mechanism for epigenetic reinforcement among mammals in this context of T cell exhaustion, at least for mice. The other important part is not just a fundamental observation. We can use this information now to try to better inform on how we treat patients. And so we tested that concept that I just said there using our DNMT3A programs and cross-referencing it with established gene expression data sets. And so here we took the gene expression data from Carl June's group, where they were looking at patients with CLL that were treated with CD19 CAR T cells, and they generated a really robust gene expression profile of just the GMP, so just the product prior to infusion, and then they read out some, responders versus non-responders, clinical responders versus non-responders. And so we asked, can you delineate the responders versus non-responders by cross-referencing this gene expression profile with the DNMT3A profile? And indeed, it was shocking. You could pick out who was going to be a responder versus non-responder quite well, simply by knowing if they had this DNMT3A program or not. And so this is great because now on the front end, even prior to infusion, you can anticipate whether there will be a response for these patients and hopefully do something for them. So we've started, this was bootstrapping the DNA methylation program to the gene expression profiles. Now, as Steve has, and with Amy's help here, got our own CAR T cell trials going on at St. Jude, we're able to access the samples directly and look at the DNA methylation profiles. And so here I'm just showing you the setup. We are able to now, and I'm not even showing you, but we get apheresis, we get GMP, and we get post-infusion products from these patients. And so it's a smaller cohort, but we're able to get really nice longitudinal data from these samples. And one of the things we found that was really interesting was actually, CD19 CAR T cells sort of the poster child, but sometimes you have responses that just, or T cells that don't give you a good response. And so we were able to kind of pick these out by looking at just directly profiling the methylation status of the cells and using that multipotency index I just told you about. So the cells that had the lowest multipotency index, the GMP product, they had the worst week 1 expansion. And of course, the ones that had the highest multipotency index, or not, they had the best week 1 post-expansion. So now, and we have a lot of cross-sectional data, but I'm just showing you the longitudinal data here. These cells that go in, they're actually pretty good, but they undergo a terminal differentiation because they're going to be exposed to chronic antigen. And you can track that very nicely with the change in this multipotency index. And this progression towards this terminal state is absolutely associated with the acquisition of these DNMT3A programs. And one of the things, there's a lot of discussion now about whether these cells, these long-lived cells, are actually functional. Are they protective? In some cases, they do look protective. And this was a drawing that at the time Caitlin Zebley kind of came in and was asking the questions. And I actually love the drawing because it's so simple, but it actually spot on in terms of what the important question is. So what she was articulating in this drawing was that when you have CAR T cell therapy, you have these CD19 positive cells. They start off really high. You infuse the 19 specific T cells. They expand. You have control of the antigen positive cells, and then you have contraction of the T cells. So we get out to this time point, and what happens if the antigen positive cells come back? What will the T cells do? Are they functional? Will they mount a recall response, or do they maintain their numbers or decline even? And so this is a tough thing to ask in patients, but what you can do is you can count the number of vector positive cells, and that's exactly what they do. And you can look at it at different stages of the therapeutic response. So during the primary response, you can indeed see this massive clonal expand, or this exponential expansion. T cells go in, this is here, and then they expand. You can see the vector copy number goes up, and this is what everyone reports. And one of the things you can do with the kids here, you can look at time points where you start to see B cell reconstitution. And so sometimes these kids will start to reconstitute their B cells if you don't get them to transplant soon enough. And what happens actually is they're CD19 positive normal B cells, high expression, a large percentage of them, upwards of 20% in the blood, but you see no change in the CAR T cell numbers. So the CAR T cells are not responding to this large, abundant population of antigen positive cells. We had a couple patients where the CAR T, they underwent antigen positive disease relapse, and that does occur. That occurs in about half of the pediatric population. It's not discussed, or it is discussed, but it's for some reason not highlighted enough. So in these two patients, you had antigen positive disease relapse. So in the primary infusion, the CAR T cells expanded. Here you see it, and boom, relapse, no change. No change in the CAR T cell numbers. Patient 6 here, antigen or CAR T cells expanded, boom, relapse, no change in the numbers. You can reinfuse, and you can see, and if you have enough cells left over, you can reinfuse. You can see some cells expand, and it's suggesting that these cells here are just not functional. So how do we make them functional? And is this approach of DNMT3A knockout a viable approach to maintain protection over long periods of time? And so we tested that in the best way we thought possible by using a serial challenge model in mice. And so here we set up mice that were infused with BB173, it's an ALL line. And here we use, and then we infuse CAR T cells with the clinical CAR, CD19 CAR. And we took the mice that had control, waited for quite a while, and they had control. And then we re-challenged them with antigen positive tumors. And this is tumor only, and the tumor grows very fast. And then in the DNMT3A knockout animals, there appeared to be protection. So the tumor could not grow. But then if you wait and you re-challenge those animals with antigen negative tumor lines, the tumor grows just fine in those DNMT3A knockouts. So this suggests that this was indeed antigen-driven protection in these NSG animals. So the DNMT3A knockout CAR T cells, at least in this setting, can provide protection when the tumor is antigen positive. So as this paper was coming out, you know, I even get stopped in the elevator now saying, oh, what are you going to do? You're going to actually knock out DNMT3A in these CAR T cells. And you're just going to give these kids more leukemia, because this, of course, we all know that this drives leukemia. And actually that's not true. And at the time, and I saw a wonderful talk from Benjamin Ebert saying the single mutation DNMT3A is, as he described it, is insignificant risk over a 10-year period. And so, as I said before, 40% survival rates at 2 years. So, and here's some data that came out from Coleman Lindsley, who came from Benjamin's group, when they were looking at individuals that received transplant with donor cells that actually had DNMT3A clonal hematopoiesis mutations. And so this was a surprising thing. The individuals that had the DNMT3A mutation in the donor cells actually had better progression-free survival. It wasn't worse, it was better, like 20% over 5 years. And this is the interesting part. When they had to treat with post-transplant cyclophosphamide, which is for GVHD and presumably to get rid of the T cells, you lost the efficacy. They lost the efficacy. So this was really suggesting that the DNMT3A mutation provided T cell based protection against leukemia. So in their paper, they had said donor DNMT3A clonal hematopoiesis was associated with improved recipient survival because of reduced relapse risk. So putting this all together, we feel very justified in moving forward with a CAR T cell protocol we're calling PERSIST, where it's going to be knockout of DNMT3A with a bicistronic vector that targets both CD19 and 22, because we do acknowledge that this provides pressure on one antigen, and there could be escape. And we'll be moving forward with this, and this is being led by here at St. Jude, Becky Epperly, Amy Talor, Caitlin Zebley, Guidra Crenchard, and Paulina Velasquez. So this is, you know, I see I'm about 40 minutes into my talk here. So this is where we're at. We've taken this concept, this fundamental observation of epigenetic regulation, controlling this sort of this developmental step here, and we're still working to refine it better. And we're using this now, though, to use genetic engineering approaches to block exhaustion and preserve this stem-like state. We're trying to hit this sweet spot to preserve the therapeutic response. There's a lot of other angles to take with this program. As you hopefully saw, we're defining the epigenetic states along this path. And this is critical for having metrics to indeed show you have erasure if you're using a reprogramming approach. And so we're obviously doing this in the lab as well. We're trying to walk these cells backwards, but if you don't have sort of these waypoints along the line of development to know where you're going, how do you know you're walking backwards? So this is another thing we're doing. And hopefully providing a good roadmap for everybody else. So this is a question I get. We have a lot of sequencing data now. Do we see better survival among cancer patients with T cell multipotency mutations when treated with ICB? So that the prediction you would start to see these individuals have better survival. And I can tell you now, the answer is yes. And so this just came out, and unfortunately it wasn't DNMT3A, but it was another driver of clonal hematopoiesis. And so what was done here with Kirsten Groenbeck and Casey O'Connell, and this is through and we got to learn of this through our collaboration with the Stand Up to Cancer Epigenetic Dream Team. What they were doing was they were taking patients with MDS, and they were treating with guanidine as a hypomethylating agent and anti-PD-1. And they found actually it did not work very well. This was the overall survival with the patients. And essentially there was a few that were long-term survivors, and a lot of them were not long-term survivors, unfortunately. But the ones that were long-term survivors, it was really interesting. And when you look at MDS, you look for all these regulators of clonal hematopoiesis, because a lot of them overlap. And what they found was they found individuals somewhat with DNMT3A, TET2, and they found ASXL1. And when it was mutated only in the myeloid compartment, there's no extended survival. But when it was mutated in the myeloid compartment and the T cell compartment, ridiculous overall survival. So this suggests that the ASXL1 mutation, which is part of that trifecta of regulators for clonal hematopoiesis in the T cell compartment, provides protection in the setting of checkpoint blockade. So we've now gone forward with setting up a program to test this, and I'm only going to show you a little bit of the data because I want to move on to something else after this. But indeed, these regulators contribute to a protective response in patients. And so this is just an experiment now where we're really trying to compare all these regulators head to head. And I'll show you, give just a little teaser of this data. And this is work done by Taegun and Caitlin Zebley, where they're taking individual regulators, this clonal hematopoiesis regulators, disrupting them in T cells and asking, do they respond to checkpoint blockade? And so we got this working pretty well. Now you take wild type P14s, these are TCR transgenic, you activate them, do CRISPR editing, and you put the T cells into a mouse, chronically infect it, wait till that late time point, and then initiate PD-L1 blockade. So this is what happens. And so this is essentially published work, although we've done it now with CRISPR. In the DNMT3A knockout, the CAR T cell, or these TCR transgenic cells, they expand really well. They undergo contraction. You wait, you hit them with PD-L1 blockade, and boom, these suckers expand really well. It's incredible. Same thing is observed with TET2, although we have some issues, and we can talk about this later. They start to expand without PD-L1, and I think that is a little concerning, and especially when in the context of the paper that Michelle Sadlin just published in Nature. And ASXL1 does the same thing. These suckers just pop really well. So they expand, they undergo contraction, you hit with anti-PD-L1, and these cells expand massively. There's some really interesting biology going on here. I'm gonna shift gears in a bit, but I'll just show you a little bit more about these regulators of clonal hematopoiesis. And as we move forward with some of these as targets to enhance T cell immunotherapy, there are things that we need to pay attention to. And so this is the T cell expansion and the maintenance of these T cells over time. We look at them during chronic infection. We also look during acute infection. So again, the ASXL1 is the best expansion, undergoes contraction, and follow the blue line, it stays flat. DNMT3A does the same thing. The numbers are always higher than the wild type. But this is a really interesting thing with the TET2 knockout. The expansion is always a little bit lower in the chronic setting, undergoes contraction, and then at this point, they start to expand. And in this model system, the antigen starts to go away around day 60, and then that expansion stops, thankfully. And in an acute setting, we don't see that. So there's something for us to really explore here. But there's a delineation between this late response in controlling sort of, I guess it's not necessarily homeostasis because it's antigen driven, and I would say it's consistent with what Michelle was saying, that there's a hyperproliferative state that's just associated with a TET2 knockout. It's not leukemic, but it is a hyperproliferative state. Okay, so what I've shown you now is that these epigenetic programs in mice and human limit the efficacy of both ICB and CAR T cell therapy. And we're working out all of these programs that control this developmental transition from a responsive to a non-responsive state. The DNMT3A knockout CAR T cell studies serve as proof of principle that you can indeed manipulate human T cell epigenetics to enhance immunotherapy. We're moving forward with that here at St. Jude. And that the epigenetic states are also predictive of the developmental potential. And all of this has led up to the development of this trial called Persist at St. Jude. And also rationale for identifying other epigenetic pathways that could potentially be targeted through erasure. So here we are, we have these therapies that are giving us these long-lived drugs. But how do you measure age? And this is where I'm at in my career. I call this obstacles and character development. It's also act 2 of hopefully my career with a lot of friends. It's also being described as having your butt handed to you by the thought leaders in the field. And so I've started to explore these concepts with a friend of mine, Dave Massapes, where we start to think about why is age relative and why should an immunologist care about aging? And I think as we've started to generate these super long-lived therapies, we have to think about how they change over time in an aging individual. Why can a Greenlandic shark live 500 years when this little human, which he could be easily eaten by this guy, only live max 122 years? What is age and why is it relative? So here's this shark. He can live 500 years. Leonardo da Vinci could only live 670 years. So in fact, we probably have sharks swimming around right now that overlap with Leonardo da Vinci at some point. And we have this little mouse. It makes you start to ask these questions. What is age? And what is it? And how do you define it? And I realize I'm running short on time, so I'll cut to the chase on this story. But I want to ask this question: is our molecular clock a sundial or is it timer? Are the sands of time ticking away, or are there scenarios where T cells can be extended? And I think a lot of people probably have already seen Dave's paper where you could continue to have these cells expand over time. And so we're asking in these model systems where you can have extended periods of aging or putative aging of a T cell, can you do this? Can you achieve cancer-free aging beyond lifespan boundaries? And these are things to think about because we're generating these long-lived therapies in kids here at St. Jude that hopefully will have another 70 years left to their life. And can we keep these therapies going? Do they age with them? And so I just want to give you a quick introduction to the aging concept and the models that Dave has introduced to us. Hopefully a lot of you have seen this paper that's out in Nature now. This is a beautiful model that Dave gave us to really explore T cell aging. And so what he's done in these studies is he's infected animals. He let those T cells rest for 30 or 60 days. He boosts them with another strain, lets them rest another 30 or 60 days, boost them again with another pathogen, another strain of the virus, and then lets them rest for another 30, 60 days. Transfers these cells now to a congenitally distinct animal, so you can really track the T cells. You're getting the T cells out aged really long now, and does it again, boom, boom, boom. Boost, boost, boost. The T cells expand, they keep going, they're just fine. He's repeated this now upwards of more than 15 times. These cells are out to around 50 boosts. This is equivalent to 2 versus 4 mouse lifetimes of a T cell. These T cells, and what he's shown now is these T cells can far exceed the lifespan of the organism. And the trick was going back, we think the trick was going back into naive animals. And so this was a 10-year running study by Dave Massapist, and I like to show this picture because it's just, he deserves a superhero pose for this. Is sort of like a Batman-like picture that Dave has, and I really think he deserves it. This was a heroic study, and has given us model systems to really explore the concept of longevity and aging in animals. So what's a 10-year running cohort look like in human terms? This is what it looks like. It's relative, right? So this handsome young man right here, it was a great 10 years for him. Look at him. He matured into this this individual here. Not a great 10 years for this old decrepit fool here. There's a lot more gray hair in the beard, a lot more crow's feet at the eyes. This gray hair might be correlated with this person starting to drive. Aging is relative, and maybe we can look at human systems, the Department of Immunology around the world, and these can serve as models of accelerated aging in humans. Although I just saw Sue. Sue, you look wonderful, so I think you're the outlier for immunologists. And I'm going to cut to the chase here. I know we want to get to questions. So Dave and I did whole genome methylation profiling on these T cells to ask, is that molecular clock resetting, or is aging at an epigenetic level persisting indifferent to the chronologic age of the host? And so what I'm showing you here is there's a global reduction in the DNA methylation profile over time. Naive T cells, and you just follow the red triangle here. These are individual mice. Naive T cells have high levels of methylation. Young memory T cells have a reduced level of methylation. And this is young, this is 90 days, sorry. These are young memory in the context of the age of the host. And if you look at about a half a lifetime, the level of methylation continues to be reduced. And now if you look at sort of non-physiological lifetimes, 2x lifetimes or 4x lifetimes, you have these continued reductions in overall methylation. This was exciting, but it was already known that aged non-malignant cells continue to reduce their DNA methylation as they proliferated. What was really interesting is that we found these discrete regions in the genome, and this is unpublished. We found these discrete regions of the genome that acquire DNA methylation. And this is associated with the age of the T cell. And not only is it associated with it, and I'll get into that now because we've made an epigenetic clock. But what was really unique about these regions is that they're all or predominantly enriched in cell cycle regulators. So you have this huge enrichment of very significant methylation at genes that control cell cycle entry in this setting where the T cells continually to be boosted. And the other important part here, and we can talk about this later, is that it's only at CPG islands. And that's the really exciting thing here. You had to have a high density of CPG sites. It's conserved across mouse and human. And so now what we have is, and here's an example. I'll give you a quick example of this. And it's in the classic cell cycle regulator molecule. It's the CDKN2A locus. I think a lot of people that study tumor biology are familiar with this. I'll just remind the immunologist what it does. It regulates. It's a classic regulator for cell cycle entry for P14, RFP15, and P16. It's set up this way, as shown by Chuck Shear and Ned Sharpless. We miss this as immunologists, though. And here's a reason why. If you look at the young memory T cells, the locus doesn't look different at all. I mean, these are CPG sites across the locus. But as you look at a half a lifetime or these non-physiological time points, the locus itself becomes completely red or methylated, except for the promoter regions. And this is just another snapshot. So this iterative boosting and aging really draws in the methylation to these classic cell cycle regulators. And we've been able to use this now to establish a T cell clock. Is actually better than the established clocks out there for organisms. And one of the things we're now that we have this tool in hand, we can ask that really simple question: is the age, or the difficult question, but simply stated, is an age of a T cell as defined by the epigenetic clock, is it associated with the age of the host? And remember, we kept going back into naive animals with this, and the answer is no. And the answer is no. And this is the predicted. This is Steve Horvath's clock of the organismal clock. And just the new T cell clock we generated. This is 1 lifetime, this is 2 lifetimes, this is 3 lifetimes worth of aging, epigenetic predicted aging. And the answer is no. We can predict these T cells to go out pretty far in their age, and it's not the age of the host. We have human cohorts, aging cohorts. We can see that the age of the PBMC does track with this. And if you pull out specific populations of T cells that we know to look at, and what I'll have you focus on here is just the CMV specific T cells from 50 to 60-year-olds. These should be about the same age as the host, and in fact they are, but the naive from the age-matched individuals look naive based on our clocks. They look like they're 0 years old. So the age of the T cell is uncoupled from the age of the host. And the important part to the story, the second story, is these long-lived, almost immortal-looking cells. They don't acquire classic leukemia-associated programs. And we have a lot of leukemia to look at here at St. Jude, and we characterize this very carefully. So these clocks show that T cells are living 4 lifetimes, and that's in the mouse, but also you can look at this in a lot of aged T cell population. Very aged and highly proliferative, but they don't undergo classic programming that's associated with leukemia. That's what I'm showing you here. And so there's a difference now. There's something that goes on between immortality versus malignancy. And that's what we're trying to get to in this new age, or this new stage of immunology, where we're looking at long-lived T cell responses in treated patients. So this last part, I rushed it a little bit. And if you have any questions, I'm happy to talk afterwards. But these multi-lifetime T cells are not epigenetically or functionally exhausted. One of the interesting things that came out of this, and this was confirmed, this was published, but then confirmed in the whole genome methylations, these stem-associated transcriptional regulators are fully repressed in these mouse and human iteratively stimulated T cells. So it makes you start to question the emphasis on these factors after you've established these long-lived cells. The malignancies associated with lymphocyte origin are highly enriched for these programs, and they're coupled to replicative senescence. They're in these classic genes such as CDKN2A to be that the tumor biologists have known about and have described to us for a long period of time. This T cell experiential age is not coupled to organismal chronologic age. I didn't have time to show you this, but we did a lot of work on human pediatric leukemias because we have access to this here. And so we can uncouple now the age of the host versus the age of the leukemia, because this works for leukemia as well. And one of the most important points here now, I think this is a question or a concept I ask a lot of people to think about is, what does this teach us about aging? If I would have asked you at the beginning of this talk to define aging, most people, I think, would have some sort of definition that talks about dysfunction. But Dave's cells actually have taught us that T cells are indeed aging, and the age is not bound by the lifespan of the organism. And the challenge is that broader concept that aging is inextricably coupled to dysfunction. So as these concepts have emerged, I think we need new tools. And we need these new tools also because it's not just for better understanding of how T cells work. We have these living drugs now. And I think these living drugs, we're hoping that they're cancer-free, but also give us an opportunity to better understand how we can learn from them and establish cancer-free aging for us as humans. Okay, I'm going to stop there. I think we take questions. I'm not quite sure how this works. Sorry, but I need to acknowledge the people. I acknowledge them along the way. This is my team here at St. Jude. A lot of the work was done by the computational work was done by Tian Mi. The new ASXL1 study is being led by Caitlin Zebley and Taegun Kang, and wonderful collaborations now from the Stand Up to Cancer team: Peter Jones, Steve Balin, Casey O'Connell, Kirsten Grombeck, and great support from St. Jude along the way. And I think we'll stop there.
Great. Thanks, Ben. That was really fabulous. Super exciting work. We don't take questions, unfortunately. We have a Twitter way of letting people ask questions. Ben is not on social media, so which is fine, which is totally fine. I totally agree with you. But the way that we will try to get your questions to you is through Twitter. So please go to Global IMMUNOTALKs, and then you'll see the place to ask questions for Dr. Youngblood, and then we will forward them to you and then put them on Twitter for people and replies. So you'll get that over the next week. So, but thank you so much. This was just fabulous. I learned so much always from listening to you. So.
Oh, okay. Thanks, Sue.
Yeah. And we'll see you all next week.
Okay.
Thank you. Okay.
Wonderful.
Bye.
Bye.